I was giving it away now, sport.
Yep. So it's my great pleasure to introduce Professor Russel Hill. It was actually a last-minute replacement because we had another speaker lined up. You might have seen the program who has had to pull out. So we're very grateful to Russel for stepping in and giving us this opening plenary. Russel completed his PhD at the University of Liverpool, if I'm not mistaken, and afterwards moved up to Durham, and where he's now a professor, and he's been at Durham continuously, I think, since moving up there. Has a long, for those who follow his work, he has a long history of work on behavioral ecology of non-human primates, particularly in relation to predation, and in addition he's worked on various aspects relating to wildlife conservation, in particular the potential conflicts between wildlife and humans as they encroach on each other's territory, so to speak. But some of you might also know his work on humans, in particular the work on the color red and its importance for contests or competitions and dominance. He's currently running the primate and predator project at Soutpansberg in South Africa. Okay. Where I believe some of the data from this talk will come from. And that leads me to introduce his talk, which is How Strange Are Your Wild Primates? Observer Impacts and Observational Data Collection. And I'll hand over to Russel.
It's almost like you must have heard me talk before if you clap at the start, because that's probably more appropriate than waiting till the end. So thank you very much, Thomas. Kind introduction, and I do feel there's something of a fraud standing up here, given the nature of the conference and what it is that I've traditionally worked on. But very recently, I have at least published on humans. In fact, I think my most recent publication was on the colour red, with Thomas actually as one of the co-authors on that, an extensive reanalysis of the role that red plays. But today, I'm going to return to what most of my research has been on, and on wild primates, but what I've tried to do is to structure it around a theme that should be familiar to everybody in the room, and a narrative that really has played out an awful lot over recent years about how we address a number of fundamental, or identify and address a number of fundamental biases in behavioural research. And this isn't a new topic. Perhaps so many of you will hopefully recognise this as being an image of clever hands, which is probably the classic example of unconscious bias, leading to a false result in animal behaviour. So back at the turn of the 20th century, clever hands was purported to be a horse that was able to count. So if its owner asked it to add 3 + 5, clever hands would tap out with its hoof 8 times, and then receive a treat, its favourite vegetables, for the successful outcome. And the initial observers of this thought that this was a genuine ability of the horse to count. But careful reanalysis by Oscar Punkst revealed that actually if the horse couldn't see its owner, it couldn't count. It was cueing in to cues that the owner was providing. And so clever hands couldn't do sums, but was incredibly good at observing human behaviour and the unconscious cues that gave in allowing the horse to arrive at the correct answer. And of course, in animal behaviour research, like human research, the goal is often to extrapolate the behaviour of a subset of individuals to that of a population or species level. And in order to do that, the subjects that we work on need to represent the broader population quite closely. And when that doesn't happen, we end up with a sampling bias, or the non-random selection of study subjects. So that you can see in this example that if we were to sample those research subjects in the circle, we've got an overrepresentation of the green and purple, a complete absence of the red and the blue phenotypes. And so we're unlikely to be able to extrapolate beyond the sample that we've recorded. And of course, that these sampling biases can lead to research that's simply not repeatable or generalizable, and to potentially false conclusions. Now, in talking to this audience, I realise, I'm not telling you anything you don't already know here. The problem of sampling biases, particularly in fields where humans are the focus of study, is really well recognised. So across the cognitive sciences, behavioural economics and psychology, the idea that results can't be generalised because the participants of those studies are described as weird, Western, educated, industrialised, rich and democratic, and therefore unrepresentative as a subset of the global population is really well recognised. So this initial paper by Joseph Henrich and colleagues has been incredibly well cited. Or if you're applying for a grant and promotion and want to use your Google citations, it's been exceptionally well cited. And while it's not without its consequences, it's created a sort of a weird versus non-weird dichotomy. It doesn't reflect the full diversity or important dimensions of diversity that exist within human populations. It has served as a critically important reminder that research is often biased towards certain demographics in psychological and other research. But what I want to move on to today is that mounting evidence suggests that there could be similar sampling biases in research on animals. So almost exactly 10 years on from the paper that really made the call to widen the pool of human participants that take part in psychological and social science studies, Michael Webster and Christian Yurts introduced STRANGE, a framework for animal behaviour researchers to interrogate and ask the question, how unusual are your study subjects? And what they asked researchers to consider is, are my animal subjects strange when compared to the wider population, for which I wish to make broader inferences? And they asked people to ask that question in one of 7 categories. And they argued that this framework should really aid the design of future studies, but also help to surface the potential biases that exist in studies, and ask people to declare those up front when discussing, publishing their completed work. And so I want to explore today the degree to which that also translates, because a lot of this the framework and a lot of the examples that are used in the paper particularly relate to experimental work in animal behaviour. I really want to explore whether we can actually utilise this framework to the degree to which it will apply in wild primates. But as I was putting this together, I also found that I got a little bit distracted, and I'm going to just very briefly take you on that distraction. I've never developed my own acronym. And I was wondering how you actually go about that to position yourself as having done something important. And obviously, in developing this, and I apologise, if you're in the room and we're involved in this, or if you're watching this on the YouTube channel, because it's an incredibly important framework. But I figured if you're starting with weird and you want to develop a very similar framework for animals, then the first thing I would do is go on to Word and ask, what options do I have for the potential, what are the synonyms of weird that I might be able to use? And you can immediately discount a couple of these because they're too short. It's not going to be important enough. And then actually a number of the others you have to discount because they've got repeated words. Unusual's got 3 U's. You're not going to be able to do anything with that. And I'd really like to think that a number of lab meetings or conversations were spent wondering, because I think bizarre would be brilliant, but what can we do for Z? And ultimately, I think that leads us to the fact that they probably had 2 options. I actually think peculiar would have been a better phrase, but clearly if you're developing an acronym, you need those letters to stand for something. So what does it stand for in the context of strange? Well, the S stands for social background, and it's recognising that animals differ in their social status, the nature and frequency of the interactions that they've had with others, and their past opportunities to learn socially from other individuals. And that might influence their performance in various experiments or the ways in which they behave. Animals also differ in their trappability and self-selection. These are two very closely related processes. They might be thought of to some extent as personality. But it means that individuals with particular traits are likely to self-select or more likely to be caught and to participate voluntarily in experiments. Animals also differ in terms of their rearing history. They will have different developmental experiences, including the extent to which they've been exposed to a stimulating environment. It may also include the degree to which they've been exposed to humans. And these experiences during development can affect potentially brain development and in turn cognitive and motor performance. Animals also differ in the A stands for acclimation and habituation. And this obviously recognises the fact that behaviour changes over time following observations, handling, tagging, or exposure to testing. And where animals sit within this process of those repeated exposures can influence the outcomes of your study. There are also natural changes in responsiveness. Behaviour can follow daily, seasonal, or reproductive patterns. It can change across development. And so we need to recognise that things are going to differ based on maybe when experiments are conducted within the year, or where in the developmental cycle an individual might be. Animals also differ in their genetic makeup, and this can have profound effects on the behaviour and the phenotypes that we might develop. And this is particularly true for species that are captive bred and taking part in animal behaviour experiments. And finally, animals might differ in their experience. And this encompasses a range of things, such as the opportunities for individual learning, perhaps participation in earlier or similar experiments. And for long-lived animals such as the primates that I worked on, they can accumulate quite complex range of experiences that could influence the way that they perform in various studies. So, since it was published, this has also been very highly cited, given the number of years that it's been published, although it hasn't quite had the Google bounce that we might expect yet in other studies. And what's particularly positive, I think, from the uptake of this is that about 15 to 20% of the citations actually relate to primate studies, particularly those working on primate cognition, primate communication, as well as cultural evolution, highlighting that within primatology, at least, there's an understanding that we need to recognise that some of these biases. But what I want to try and do over the remainder of the talk is to give a sense of how these biases might relate to studies of wild primates, drawing on my own experiences, but also highlighting then some of the areas where we might need to extend the framework. And if I'd had enough time thinking through acronyms, I might have tried to add a letter to the end of it, but I haven't quite got that far. But I want to show some areas or some things that aren't really captured within this that I think are also biases within the data that we collect. So, if I take you back quite a lot of years to my PhD in Liverpool, and when I went out to do my own PhD fieldwork on baboons, and even then we knew about some of the sampling biases that occur within primate behavioural data collection, and particularly here in the context of social background. So back in 1982, Martin Sharman and my PhD supervisor, Robin Dunbar, had published a paper looking at the selection of study groups across a range of baboon studies. And what they found is that researchers tend to select the largest group available within a population. And this was particularly true. Now, what sort of pointer are we working? Okay, so this was particularly true in those populations where the maximum available group size was 85 or less. So the figure here depicts what the ratio of the study group to the maximum size of group that was available. And where group sizes within the population had a maximum of less than 85, actually the majority of the study groups that were selected were that largest group available. And almost all of the study groups selected for observation were at least 80% of the size of that largest study group, and all of them over 50%. Where the maximum group size within a population was larger, you do start to get some variability, but nevertheless, still the most common group size was the largest group within the population. And even though there are some groups that are less than 50% of the group size, it is still the case that most of the studies were selecting a particularly large study group for observation. And of course they were doing this to maximize the sample sizes of the data that they were collecting. But in doing so, particularly in those contexts where only a single group was being studied, you were reducing the opportunity to generalize from the results. Now, I was fortunate to know about this before I went out to collect my PhD data, and fortunately the design of my PhD required me to have one large and one small study group. So I overcame some of the challenges inherent within this. But I was lazy, and what I did was just simply selected the two groups closest to my house, and therefore introduced another level of bias within it, was that I'd chosen those with a much greater exposure to humans and could potentially much easier to habituate. But fast forward then to one of my more recent PhD students, Ben Walton, who's been, or as part of his study, we adopted the increasing trend of using technology to explore the fine-scale detail of animal behaviour. So here a GPS collaring study, where the GPS was collecting very high resolution, 1 second resolution data that allows you to plot the fine-scale movements of baboons in a farming landscape. And here we were really trying to understand the patterns of crop foraging by the baboons in the environment. So I'm not going to go into the details of the results of this study, but often people advocate for this sort of technology as a means of overcoming some of the observer biases that are inherent in the ways that, as researchers, we collect behavioural data, and something that I'll explore in more detail at the moment. But nevertheless, even though it can help overcome that, it can't overcome some of these, the trappability and self-selection problems that we have. So, quite successfully managed to trap and collar 19 individuals across the course of 2 field seasons. And there's a practical reason why you have to bias your collar deployment to adults, because they have to be heavy enough body mass to ethically carry the weight of collars that we're deploying. But nevertheless, you can see that even when we've done that, what we've predominantly captured are males. Both, or almost entirely subadult males, were all male. The majority of the adults that we were able to collar across the 2 seasons were also adult males. Now, sometimes this is because we avoided deploying collars on pregnant or females that had young infants. We released those immediately. But nevertheless, there's an inherent bias in the data that we were able to collect, because the males are simply more likely to wander into the traps. And in fact, sometimes they will wander into the traps the day after you've trapped them as well, because there's food inside, so why wouldn't you? So this obviously then limits our degree to generalize from those sorts of data, and we have to be very specific about what it is that we are able to look at and what we're not able to look at. Now, I could review each of these various components of the strange framework in turn, and we can find ways in which while primates, or there are considerations about the biases that we may be inherent in our data collection in relation to wild primates. But I particularly want to focus now on the remainder of the talk on the fourth of these components. That's the acclimation and habituation. And as a reminder, that's really looking at how behaviour changes over time following observation, handling, tagging, or exposure to new experimental testing situations. And where animals sit within this can influence the study outcomes. And in particular, I want to just focus on the observation component of it, or that's really where I'm going to focus much of my attention. And in doing so, that leads us into another form of potential bias in data collection that's also long been recognised in many disciplines, particularly in the behavioural and social sciences. And that's that the very act of measurement or observation directly alters the phenomenon under investigation. And there are two kinds, effectively, of observer effects. The first is that the presence of an observer influences the behaviour of the study subjects, in our particular case, animals or primates. This is sometimes called the experimenter bias, or, depending on the discipline that you work in, a Heisenberg effect, a Hawthorne effect, a Rosenthal effect. It's well recognised in various disciplines. The other type of bias is that observers can subconsciously influence the data if they have expectations of the results or outcomes, sometimes known as a confirmation or expectation bias, and something that within psychology and other fields is well recognised, but can have within animal behaviour potentially important implications in terms of the way that we collect data. And of course, these two types of bias are not mutually exclusive. Indeed, the unconscious behaviour of the observer can also influence the animal's behaviour, so that the animal adapts its behaviour to the observer's expectations. The clever hands effect. But I want to focus particularly on, or predominantly on, the first of those, and the observer effects, and how being an observer in a system influences the behaviour that we record. And in 1948, Kinji Imanishi began a study of wild Japanese macaques, where he established a set of methods, habituation, individual recognition, and long-term study, that have become the foundation for behavioural research, particularly on primates. And over the last 60 or 70 years, these methods have become embedded in some of the longest running and well-known primate field studies. Jane Goodall's long-term study of chimpanzees in Gombe, Stuart and Jean Altman's still ongoing Amboseli baboon project. All of this, well, and actually throughout primatology, I think, research using habituation is almost ubiquitous. It's the standard method that we use. But it extends much beyond primatology. The Kalahari meerkats have been subject to long-term research following habituation. Studies on birds, such as the Vulturine guinea fowl project, also use habituation. And all of this is predicated on allowing you to get close to your study subjects so that you can accurately record the behaviour that you see. But what we find is that the implications of this behavioural choice, or this methodological choice, its impacts on behaviour or the data that we collect, are barely recognised. And I want to question whether that is actually reasonable, particularly in the context of the strange framework. And so I'm going to talk to you about some of the primate behavioural studies that I've worked on, drawing on some previous postdocs and PhD students that I've worked with, and how we can chart the changes in behaviour the longer that we work with our study animals. And two of my PhD students here, Pete Tomlin, collecting data on a group of baboons, Andy Allen, whose work I'll draw on quite a lot towards the end of this talk, working on those same baboons 10 or 15 years later. Both of whom worked on habituated, or helped initially habituate and continue to support the habituation of those animals so that we could collect data from close quarters. So what do I mean when I talk about habituating the animals? If you look into the primatology literature, you can find a wide variety of definitions for what habituation means in the terms of primate data collection. But ultimately, it's about your study subjects accepting humans in close proximity, apparently ignoring them, so that when you are alone with your study subjects, the study subjects are calm and you're able to collect data from them at close quarters. And within this particular definition, which is taken from a study of chimpanzees, you get a sense of the fact that what we're trying to do is just to become a background part of the primate's environment, but in doing so, it's particularly about allowing us to collect good quality data while these animals move around their natural environment. And in essence, these sorts of definitions embed two key assumptions. The first, then, is that we do become a background part of the primate's environment. We're a neutral part of their environment. But there's also an implicit assumption that within the social groups of primates that we work on, that all animals are equally habituated. But rarely are these assumptions explicitly mentioned, and very rarely are they actually tested. So what I want to present to you is some evidence from the work that we've done over recent years. And as Thomas mentioned, my particular interest has been in studying predator-prey interactions. Not so much the direct interactions and the direct mortality that occurs, but understanding the fear that is generated by having predators in the environment and how primates respond to this fear of having predators in the environment. And if I know what I know now when I started on this, I would have probably taken a different approach to addressing some of these questions because, we just mess up the systems we work on, and it actually makes it very difficult to address these sorts of questions. And actually, maybe if I'd read more widely, I would have already started to get this at the beginning, because actually we've known for a while that primate research influences predator-prey interactions. Lynn Isbell, back in the early 90s, published a series of papers from the Behaviour project, and she was able to show it's that across a range of study groups, the further those study groups were from their research camp, the higher the predation rates that were experienced by the groups of monkeys. To the extent, these are really quite crippling levels of predation. Nearly 80 to 90% of the population was being taken each year, predominantly by leopards. But those close to the research camp seem to be afforded a degree of protection. And in a particularly nice anecdote is a phenomenon that became known as the Nairobi effect. So when the Amboseli Vervet project took a little R&R, went to Nairobi to enjoy the nightlife, they left the research camp for 1 to 2 weeks. When they came back, they discovered that a lot more vervet monkeys were missing than in any normal 2-week period. So what was happening is that the people were departing the system, and in doing so, the leopards could return to the system, and, they were purported or thought to return to the system and were able to predate upon the monkeys that were there. But I ignored that. And off I went to South Africa. My PhD was something of a failure in this domain because I went to a study site that didn't really have a resident leopard population. But in moving to Durham and to the research fellowship, I was able to establish a new site in the Soutpansberg Mountains, up in the very South Africa's northernmost mountain range in the very northeast of the country. It's not a traditional African savanna. It is a mountain range, very rugged topography. But it's home to all 3 species, diurnal species of South African primate, actually all 5 species of South African primates. And over the years, we've conducted studies on the semi-terrestrial vervet monkey, the arboreal samango monkey, and the terrestrial baboon. And in terms of what you need to know about the study site, it has an intact predator community, so a reasonably high, in fact, when we started there, one of the highest reported leopard densities in sub-Saharan Africa. Breeding pairs of crowned eagle and Verreaux's eagle, as well as predatory snakes, which is the python. And importantly, in the context of the work that we wanted to do, the humans that interact with these predators are benign. They're predominantly researchers within this mountainous environment. So very few negative interactions with people. What I was interested in trying to explain is, and we started, the initial study was on vervet monkeys, is why here is a depiction, a map of a vervet monkey home range. Some areas are used really intensively, and other areas are used not very much at all. And to what extent can predation help us explain that? And primates are a particularly good study model for this, because particularly the vervet monkey classically gives acoustically distinct alarm calls. And so you can map out where these alarm calls are to different predators across their home range, and that allows you to develop these predator-specific landscapes of fear. It's where they feel at risk from different predator types. And we can ask the question, to what extent do these landscapes of fear help us explain where the vervet monkeys go and do not go, and how does that compare to other really important resources like food availability or access to sleeping sites? And when we do that, and we've done it across all three of the study species, we've adopted the same sorts of approach, we found that vervet monkeys are really scared of leopards, pretty scared of baboons. Samango monkeys, pretty scared of eagles. That's all of these are the most important drivers of their species. Baboons are avoiding other groups of baboons. For all three species, avoiding these risks is far more important than where the food is. So you avoid the risks and then you forage. But it might also be important, and I'll come back to this, that this was also the order in which we conducted these studies. So this is the first one, the vervet monkeys in the first year that we were based at the site, or the first couple of years. This is after about 5 years, and this is using data after about 10 years of our researcher presence at the field site. And I'll return to that in a minute. Nevertheless, from those sorts of results, I was able to say that predation is really, really important. And it is, but we started to get signatures of really quite funny things happening in our data. So in addition to conducting behavioural studies, we've also tried to use experimental approaches as well as some of the GPS technology to try and use complementary methods to get at this issue of predator-prey interaction. So one of those was conducted by a postdoc that I got to work with, Kate Nowak, and we conducted a giving up density experiment. So these are feeding experiments, highly sophisticated, a washing up bowl with sawdust and peanuts in it. And what you do, if you've got a spare afternoon or two, is you chuck them up trees. For the arboreal Samango monkey, and it's basically a depletable food patch. So if a monkey approaches this food patch and starts to forage, it can find the first peanut quite quickly, and its success in doing so, the diminishing returns mean that it takes longer and longer to find the next food item. If you feel safe, you're willing to deplete that food resource quite extensively. But if you feel at risk, and we predicted that for an arboreal species they would feel at risk here on the ground, your giving up density, the point at which you depart the food patch, comes much earlier. There's more food remaining in the food patch. So we predicted then that they should feel safer the higher you get into the canopy. The highest giving up densities in these feeding experiments should be at the bottom. And that's exactly what we found. So across the two study groups that we conducted this experiment on, more peanuts remaining, a higher giving up density on the ground, and as you move into the canopy, the giving up density declines. But I still remember the day that Kate came into my office at Durham and said, "There's something funny going on there. get different results depending on whether we're studying them that day, conducting behavioural follows, or just running the experiment. Because on the days that we were following, following the monkeys from dawn to dusk, collecting behavioural data alongside this experiment, monkeys just felt safer. The giving up densities for both groups were lower when we were present as researchers than when we were not, to the extent that for one of the study groups, they didn't care whether they were on the ground or higher in the trees. They just felt safe. And it's not that leopards weren't around during the periods that we were conducting the study. They'd come and visit the experimental setup at night when we weren't there. But during the day, us, as researchers, was clearly giving a cue to the monkeys that you can feel safer. We were acting as a human shield. And this is something that's been reported in a variety of other contexts. The most famous being following the re-establishment of many predators within the Yellowstone ecosystem. So the human shield effect was first reported for the impact that recolonizing bears had on moose. And the amazing thing about moose is that they can give birth, or 90% of the females give birth in a very short time window. So in the top graph here, we've got the days across years, and this gives you a sense of the range of when 90% of the births are occurring. Just over a 9-day window, 90% of the females are giving birth. And during that period, therefore, it's quite easy to track where those females are going to give birth. So across a 10-year period, from 1994 to 2004, the dashed line here shows the increase in the bear population that occurred over that period. Bears are scared of humans. And so they stayed away from the roads and other human infrastructure. And because of that, during that birthing period, the distance of the females where they were giving birth declined, and they got closer and closer to roads or other human infrastructure in order to give birth. They were using the presence of humans as a shield against the potential risk of the bears predating on their young. The females that didn't give birth that year, or the females that were in areas that had not been recolonized by bears, they showed no change. This was an adaptive shift of those females to move into areas that they knew they were less likely to encounter bears. So it seems that a wide variety of animal species, but certainly primates, can tap into cues of how we minimize their potential risk of predation. And that also seems to have physiological impacts on the animals themselves. So another former student I work with, Laura Labage, conducted a physiological analysis to see what impact encountering predators had on their fecal cortical metabolite responses, their stress level responses. So when the number of group-wide alarm calls, a strong proxy, I think, for the number of predator encounters increased, on days where it increased, the physiological response, the stress response, was much greater, a fairly linear correlation between the number of alarm calls and the elevation in stress levels recorded in those animals. But this is the situation where the monkeys are being followed by a single observer. But on other days, we may be following them with 2, 3, or 4 observers. And as soon as the number of researchers within the group increases to 3 or 4, even though they're still giving the same number of alarm calls, there's no associated physiological response. Again, it's like the monkeys know that it's safer, it's nothing to be worried about because of the deterrent effect that we potentially have. So we started to consolidate some of these ideas into a review to try and capture the various ways that us walking around in the middle of a primate group could influence the behaviour that we record. Because when we habituate primates to observer presence, we only habituate our own study species. We don't intentionally habituate other species of primate that may be co-inhabiting with us,
or other species that may form associations. And so we're less likely to record these associations through observational methods. If we deter certain predators, such as the leopards, so I mentioned that these were really important for vervet monkeys at the start of our study, but by the time we got on to Simango monkeys, these were not particularly important in determining their landscape of fear, but other predators, such as the eagles, that have much larger ranges, are much less likely to physically interact with us or use the same plane, may stay around in the community much longer. So it changes the type of predator exposure the animals get. That in turn can change the vigilance or behavioural patterns that you record, which in turn can actually lead to some of these animals using riskier habitats, such as the Simango monkeys coming down and foraging much more on the ground when we are present. So all of this then is a significant source of bias, and one that's very rarely recognised, but is also heavily contingent on how far into your study that you are. So, I mean, it's very difficult these days to envisage a natural environment where that's untouched by human presence. But nevertheless, if we think of as close to pristine environment as we can, at the start of the study, the observed predation rates, the ability for predators to impact upon the study animals is likely to be quite high. But as we start to habituate them, those predation rates, because of this human shield effect that we provide, are likely to decline. And if the predators themselves never habituate to us, you end up with a situation where your study is conducted where the risks to the study animals are significantly lower than those that were present before you began your study. But it may be that some of the predators start to habituate. And I was talking last year, bumped in at two different places to Cathy Crockford and Roman Wittig to discuss the situation of the Thai chimpanzee project. And recently they'd been reporting they'd been losing a lot more of their chimpanzees to leopard predation, as the leopards, it seems, have started to habituate to the presence of human observers, or more specifically, they've recognised that the human observers, the researchers, are not a risk, in contrast to some of the other humans that they will encounter in other parts of the forest. So the research area is now a safer area for those predators. It's brought them into that area, and they have started to predate more of the chimpanzees, which is possibly a more natural level of predation. It may be also that some predators start to cue in to the researchers telling you where their food is. And actually you could lead to a slightly problematic, ethically speaking, situation where you increase the predation that is observed within your animals. So thus far, all of this really points to the fact that in terms of the ecological communities in which these animals are situated, we are not a neutral part of the environment. And I'll touch on that again with the final examples that I'm going to look at. But it's clear that we're influencing interactions between primates and their ecological communities, their predators, other primate groups, other species in various ways that are often unrecognised, definitely underreported, and also very hard to quantify. But I also want to just spend a little bit of time looking at actually this question of whether habituation is equal across the group. And if we assume that behavioural methods do allow us to get high quality data, can we assume that all of the animals are responding to us as human observers in the same way if they become habituated? Spoiler alert, but no is the answer to that as well. So this is coming back now to Andy Allen's work, and Andy took an experimental approach using flight initiation distance methods. So these baboons are what we would call in the literature fully habituated. They've been studied for 10 or 15 years. Many of those individuals have been born and grown up with human observers collecting data. at least on a number of days a week over multiple weeks and months a year. So flight initiation distance methods are often used to measure predation. You effectively walk towards something and you wait until it gets uncomfortable for them and they run off. And we recorded two things. The visual orientation distance, and Rebecca did a very good job there of not panicking, and it was quite late at the point at which visually oriented towards the threat that comes at you, and then also the distance at which the animal moves away. And we recorded a lot of contextual information around these repeats, and Andy managed to conduct these approaches, 24 replicates across all non-infant individuals within the study group, an incredible amount of work in order to collect these data across 2 study years. And we can make a number of predictions about what we would expect if we were neutral, if we were treated as a predator. And often these flight initiation distance methods are used as a proxy for measuring predation. And what we actually found is that the strongest support really came for the human observers being a high-level social threat. We're effectively integrated into the social networks. We're a dominant baboon as we're wandering around collecting our data. And this has really important implications. But if we consider the quantified distances at which the animals looked at us and at which they moved away from us, there are a number of contextual factors that influence the distances that we record. What behaviour they were involved in at the start, the habitat type, the number of neighbours that they had. But of all of these factors, if we look at it statistically, the strongest predictor of visual orientation distance and flight initiation distance was the individual ID, the animal that we were walking towards. So individual ID was the most important factor. And both visual orientation distance and flight initiation distance were therefore distinct between individuals, but also consistent and repeatable, consistent across years and repeatable within individuals. They were also very highly correlated. That's what's being demonstrated here. So the visual orientation distance of an animal was highly correlated with its flight initiation distance, even if we looked at entirely independent data sets. And alongside a number of other tests, this ultimately suggests that what we'd uncovered was some form of tolerance trait. Individuals varied in their tolerance of human observers. And this wasn't determined by age or sex. So this graph here, the more tolerant individuals are up at the higher values, and you can see that while we've got, the less tolerant individuals are in the higher values in this particular example. And whilst adult females occupy a lot of these positions, if we look at the most tolerant individuals, it's a combination of adult females, of juveniles, and some adult males. And perhaps the bolder individuals that we would expect, the males very often, they're actually nestled in the middle of this spectrum. So these characteristics are very much individual based. They're not determined by body size, age or sex. But they do have really important implications for the data that we collect. So many animals would be happy to be in close proximity to the observers, but perhaps you can see that some of these animals, even when they're in close proximity, might be a lot more wary of their human observers than other individuals. What implications does this have? Well, we can look at the impact that observer tolerance has on the probability that a particular animal appears as the neighbour of a focal study subject. So I'll talk you through these figures. So the 2 rows are just to show that the results are very similar across 2018 and 2019, so highly repeatable. And the y-axis is the number of times that individual appears as a neighbour in the focal samples of other individuals. And the x-axis is the tolerance of the individuals. These are the most tolerant individuals, these are the least tolerant individuals. And we basically record whether that neighbour is within a 5-metre buffer, so actually close but in a reasonable buffer around the individual, or whether they're touching. So these are likely to be two grooming individuals. And these are often two metrics that are used to develop social networks that are used in a wide variety of contexts in animal behaviour, transmission, to look at transmission chains and a variety of experimental approaches. So we've got two conditions. One is whether the observer collected the data standing quite close, and those are in the yellow, and one where the observer was quite far away when they were collecting or further away when they were collecting the data. The cutoff here of 4.5 metres was just the medium within our data set. So if I start with the blue, first of all, where the observer was far away, the probability of being a neighbour, or appearing as a neighbour in a focal sample doesn't change. Tolerance has no impact on whether you appear as a neighbour. But when the researcher is close to the focal animal, you start to, within the 5-metre buffer, you start to preferentially oversample more tolerant individuals. You undersample less tolerant individuals in these grooming networks, and that's consistent across both years. And if we look at what that means in terms of the measured social network, can you see that? Actually, I tried to do a higher contrast version. There we go. So to give you a sense of what that looks like, here we've got the two grooming individuals. This is when the data is collected close by. This is when it is collected further away. These are the measured social networks within 5 metres when it's collected close by and when it's further away. Fundamentally different social networks that are being recorded, and that would change significantly the inferences you would make about the spread of information, the spread of disease across these networks. Because this 4.5 metre distance will also change as animals across the course of a study become more habituated and maybe more tolerant of you over time. It also means that comparing social networks between 2 years of an ongoing study could also be subject to exactly these same sorts of biases. And the reason for that is that when you collect the data far away, it allows other individuals to be between you and the focal individual at a distance at which they feel happy with the level of tolerance. Well, their tolerance, you're not within their tolerance threshold. But as you move closer to collect your data, you start to displace some of the individuals that are between you and the focal animal. Now, it might be, in these larger buffers, that you only displace them to the extent that they still appear as a neighbour within the data that you collect. But where we're looking at either touch buffers or much more refined buffers, individuals within 2.5 metres, your presence focusing on this individual could displace some of their neighbours, and they are no longer recorded in the data. So what this means is that, taken together, researchers cause massive behavioural changes in the study animals that they are trying to record the behaviour of, which we cause changes in the ecological communities that our study animals inhabit, changing their interactions with predators, with potential competitor groups. And the nature of these changes is also likely to vary over time, depending on where both the study animals are, but also where the community around it is in terms of habituating to observer presence. And this can really have some quite profound long-term impacts as well. So one thing that I've not touched on, but we think is potentially important, is it can have impact, potentially long-term genetic impact on the study animals that we're working with. So this is a figure that's taken from a paper in 2020 of the Inkawu Vervet project in KwaZulu-Natal in South Africa. And this study was looking at the transmission, or the behavioural mechanisms involved in the transmission of information between groups, and whether immigration of males was a particularly important route to bringing in new innovations. Particularly in relation to
how they approached novel foods.
So the purpose of the study, I'm taking this out of context in using this figure. But what it demonstrates is that across the 5 study groups, that's a good study, good sample size, all of the emigrations into the study groups were from animals in other studied groups. Even this individual that appears to have come from outside came from another study group. It just emigrated through, or immigrated into another group before moving on and returning to one of the study groups. And when you think about it, it's obvious. If you are subadult, male, vervet monkey, you've got 2 groups to choose between, one of which has got these stupid humans, these threatening humans wandering around in the middle of it, or there's another group. Where do you go? It's only those that have got that experience of having researchers and potentially learning some of the benefits of having researchers that are likely to immigrate into a study group with researchers in it. And anecdotally, we've seen exactly the same thing with the baboons that we study. Andy's data collection for the study that I've just reported was pretty intensive. 4 or 5 days a week, there would be often Andy and maybe 1 or 2 of his field assistants with the baboons. And over a 3-year period, we had no immigrations of outside males into that group. And actually, the subadult males didn't leave that group either. Partly, I think, because there were likely to be reproductive opportunities within their natal group. He came back to the UK for 5 weeks. It was our own Nairobi effect. This was the London effect. He came home, saw his parents over Christmas, went back 5 weeks later. There were 3 new adult males in the group that had emigrated in that period where the researchers were away. And finally, it's also been hypothesized that these sorts of habituation changes could ultimately lead to changes in the phenotypes that are expressed within our study group. So the context here, so Sadul et al. Hypothesized that when human presence protects prey from their predators and the animals start to habituate to the presence of humans, then it decouples commonly associated traits related to how animals cope with their environment and the stresses in their environment, leading to the emergence of new phenotypes. Now, the context in which they were thinking about things are about how animals increasingly adapt to living in anthropogenic environments, whether that be through tourism, through urbanization, potentially even through domestication. But the context in which, the lower of the figures here, there is a strong human shield effect and habituation to the presence of humans are entirely analogous to our research scenario. And so if we focus on those, and I'm not going to explain the figures in detail, in part because I found it really hard to get my head around it, and if I try and explain it to you, we're all going to go away incredibly confused. But ultimately, what they tried to suggest was that we have a spectrum of reactive and proactive individuals. And preactive individuals are characterized generally by, they tend to be more exploratory, they're much bolder, characterized by high aggression, relatively low levels of stress. In contrast, the reactive individuals tend to be quite shy, not exploratory, have a very high stress response when things go badly, but are not particularly bold and not particularly aggressive. And what they argue is that these suites, these behavioural traits and physiological traits, the traditional patterns that you see get decoupled under conditions of habituation and the human shield.
So where there's a strong human shield and habituation, the individuals should become characterized by low aggressiveness and physiological stress responses that have enhanced behavioral plasticity, boldness, and cognitive abilities. So things that were traditionally separated within this character space actually become aligned in what they call these pre-active individuals, because it's effectively a merger of the some of the more positive elements in many respects of the reactive and proactive spectrum. And what this means is that for some of those long-term studies that I indicated at the outset, the phenotypes that we are encountering now, their responses and the behaviours that we're recording, through the selection and the presence of us as researchers over many years, could be very different and give you very different outcomes to the behaviour of those primate groups that were present at the beginning. And so to finish, does habituation cause strange animals? Do the methods that we use cause strange animals? Absolutely. Pretty much on every domain, the methods that we've chosen to use will in some way lead to the sorts of biases that we want to recognise as part of this strange framework. And in terms of trappability and self-selection, what I've just demonstrated is that more tolerant individuals are going to appear more often in the behavioural samples that we collect. But you can come up with examples for virtually all of these about how that method that we have chosen to study primates is going to influence the behaviour that we collect. And we should report that, and we should accept that, rather than treating habituation as a means to an end. So even when animals are fully habituated to human presence and are highly tolerant of humans, researcher presence then is going to influence not only the behaviour, but the communities around them that we're trying to study. And in doing so, we need to get beyond this idea that habituation is a means to an end, or it's one of those barriers to overcome. If I want to study primates, I've got to habituate them first, and then I can get on with the important stuff, because actually having made that decision, you've already changed the system quite substantially that you're going to study. But up to now, the way in which habituation has been reported is I habituated by animals, and then the fun stuff began. And we've got to move beyond that and start to think about actually what are the biases. We've got to find ways of not only reporting and recognising the way that we collect data might lead to biases, but also actually measuring and understanding the strengths of the effects to determine whether any of those are likely to be important to undermine the inferences that we've made. So I apologise, I've whittled on too long, but that's because I kept control of the stop cards. But thank you very much for humouring me.